Hello everyone, welcome back to my YouTube channel. If this is your first time here, please subscribe and thank you so much for coming back. You can leave me a like if you like this video. And yeah, let's get started. So in this video, I will be introducing you guys to purines and pyrimidines. Like I said, before I get into any explanations, I'd like you guys to understand why you are studying a particular topic and why exactly is it relevant. So before we dive deeper, I'd like to take you guys back a bit further. And I always say this: if you have not understood how to draw each and every single one of the 20 amino acids and really understood what amino acids are and formation of urea, please go back, because this video is a bit pointless if you really don't understand that basic information. So let's get right into our discussion. So I would like you to think of this, okay? Look at this house here. This house was made by bricks, and these bricks were held together by cement, okay? But these bricks did not appear from nowhere per se. These bricks in particular, we had to mash a bunch of little stones together to actually come up with this brick, okay? Now these little stones were equally made from something, and they were made from what we call atoms. So taking you backwards, imagine this protein. This house is a protein. The building blocks of proteins are amino acids, and amino acids are held together by peptide bonds. So think of the cement that holds the house together as peptide bonds. Now these building blocks of proteins, or rather these bricks, were made by stones, as I said earlier. And these stones in particular, what are they? These stones are DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And then finally, if we go a little bit further down, okay, these stones were made by atoms. So what am I trying to tell you? Why don't we start from the bottom? Atoms come together to make these little stones, okay? And these little stones come together to make bricks. These bricks are held together and come to make a house. So going backwards again, we said amino acids are the building blocks of proteins. Now, the building blocks of proteins, follow me very carefully, are amino acids. The same way proteins have their building blocks, amino acids have building blocks. And the same way DNA and RNA are building blocks of amino acids, the building blocks of DNA and RNA are the purines and the pyrimidines. And when we look at this very carefully, again, the building blocks of purines and pyrimidines are the atoms that make up the purines and the pyrimidines. So what I'm trying to tell you, at the end of the day, the study of purines and pyrimidines is basically how we are going to arrange atoms together, how we are going to build atoms from scratch, what we call de novo synthesis. Okay, we will make them from scratch. After we make them from scratch, we will make the building blocks of DNA and RNA. After we make the building blocks of DNA and RNA, we will now make amino acids. Once we make amino acids, we'll put them together with peptide bonds and finally make a protein. So guys, the same way we built our house, let's try to build a human being before we get into depth of what each and every single meaning of the topic of purines and pyrimidines actually is. So we're going to take our atoms together, okay? We're going to take these atoms, put them together and create purines and pyrimidines. So atoms are the building blocks of purines and pyrimidines. Now, purines and pyrimidines in turn are the building blocks of the nucleic acids, DNA and RNA, which are deoxyribonucleic acid and ribonucleic acid. DNA and RNA in turn are the building blocks of amino acids. So when we take three nucleotides together, and nucleotides are basically purines and pyrimidines attached to a ribose sugar attached to a phosphate group. When we take three of them, we make what is known as a codon. And this codon, as the name implies, codes for amino acids. And when these amino acids come together, according to functionality, we come up with a protein. Now, we can come up with different proteins. We can come up with muscle tissue, brain tissue. We can come up with blood. We can come up with heme. We can come up with etc., etc. There's so many things. Muscular tissue. Already mentioned muscular tissue rather, but to going deeper, you can come up with cardiac muscle, So we want to find out how our body takes these atoms together, arranges them very nicely to come up with different organs such as the brain, the heart, the lungs, etc., etc. For your basic understanding, you have to think of it in this particular manner. See, if there is something wrong with the atoms, there will be something wrong in the individual. If you don't build a proper foundation for your house might be a bit slanted to the left or to the right. It might be very weak. See, think of it like this. If when you are studying the glycolytic pathway, the only thing you studied is how our bodies can use glucose for functionality, how we can use it as a source of energy, etc., etc. You didn't necessarily care about the arrangement of the atoms, how they came together, whatnot. You didn't really care. The reason is that because we are not made of glucose per se. We are made from amino acids. So we want to find out how things go wrong in order for us to either prevent them from going wrong, or if they do go wrong, is it beneficial to us? So I'd like you to think of it like this. If you see someone with three eyes, okay, it will be weird at first, a bit creepy, and maybe you wouldn't want to try to come off like you're uncomfortable, but naturally what human will come up like we will be uncomfortable. As a doctor, you will look at them and say, okay, this person has got three eyes. How did you end up with three eyes? Is the third eye functional? If it's functional, is it beneficial to you? If it's not beneficial to you, should we remove it? And will it cause you harm if we remove it? And if we remove it, is your life better or is it worse? And also you want to find out, how did this happen to begin with? The reason why you're asking these questions is to either, number one, help the said individual in the situation that it shouldn't happen again, or to make their life better, or number two, to prevent it from happening again. I guarantee you that 99.99999% of the time, there is something wrong with how the atoms have arranged themselves in order for it to manifest in an individual when you see certain illnesses in a person. The best example is sickle cell disease or sickle cell anemia. In sickle cell anemia, the amino acids glutamate and proline are swapped, which affects the hydrogen bonding. Now, this affecting the hydrogen bonding is why they've got sickle cell anemia. Okay, and at the end of the day, where did this defect come from? This defect came from the amino acids arranging themselves. And how did the amino acids defect come about? The defect came about with the atoms. And this is why we study how to build amino acids from scratch and how these amino acids come together to make a protein. Remember, even when you're eating your chicken, your enshima and vegetables, chicken is your protein. If you're eating cow intestines, the chicken and vegetables, the intestines is your protein. Even your intestines are protein. So your amino acids will code, okay, how do we make intestines? How do we make a heart? How do we make blood vessels? How do we make a brain? Those little defects, when you swap an atom from left to right or right to left, it has detrimental effects. And this is why we study purines and pyrimidines. In particular, the one that is of greatest interest is de novo synthesis, meaning let us literally make an amino acid from scratch by getting each and every single grain of sand. We will study each and every single grain of sand and put it together to make this brick. That is what we are trying to do. So guys, I hope this was a very nice brief introduction into purines and pyrimidines. In the next video, I will talk about basically the uses and functions of DNA and RNA. I will not get into detail about the structures and what have you for the next video, but the video afterwards is when we'll now start to look at each and every single pointer, from de novo synthesis to codon arrangement and everything else. So I'll be very systematic how I deliver the information. For now, this introduction should take you through, and in the next video, like I said, it's gonna be about DNA and RNA, why they're different, but they have the same building blocks. So what makes them different, and why are they different, and how are they different in functionality? And then from there, we are going to look at the structures that make up DNA and RNA. And then now we'll begin to synthesize de novo. Thank you everyone for watching, and I hope this video was very helpful, and I will see you in the next one. Bye.